Hi, my name is Robert Krueger. I'm the research leader and curator at the USDA-Agricultural Research Service National Clonal Germplasm Repository for citrus and date, located in Riverside, California. Citrus is a crop that is widely planted in all areas with a suitable climate and soil conditions, possibly one of the most widely planted perennial crops in the world. Citrus is a crop, like many other perennial crops, that has what we call systemic diseases. These are generally diseases associated with viruses or quote-unquote virus-like particles that are generally confined to the phloem. An example of this is the citrus tristeza virus. And this virus, or specific variants of it in the 1930s and 1940s, resulted in huge losses of citrus production worldwide because the rootstocks used at that time were susceptible to this virus. Well, many other viruses are similar to citrus tristeza virus in that they are initially present in the phloem and then become systemically distributed throughout the entire tree structure. How do they get into the phloem? There can be a couple of different ways. Some of these are transmitted by insects with piercing, sucking mouth parts. Virus being present in the gut of the insect is injected into the phloem when the insect inserts its proboscis into the phloem to feed. Another important way it's transmitted is by humans. Citrus and most perennial crops are propagated commercially by grafting a scion, the usable part of the tree, including the fruit and the rootstock. And these two distinct genotypes are joined together by the grafting process, and there are various variants of the grafting process, and through wound healing become one tree with a continuous vascular system. And so if we take the scion that we're going to use in the grafting process and the virus is present inside the phloem of that, then it can become established in the tree, potentially causing large economic losses. And the same thing is true of various other types of perennial crops, as fruit and nut trees, grapevines, etc. Well, because citrus is such an important crop worldwide, and because of the potential destructibility of these viruses, which can be graft transmitted, there are usually extensive phytosanitary regulations in place at the federal and sometimes the state level of important citrus producing countries. In the case of the United States, citrus is what we call a prohibited commodity, and new varieties need to be introduced to the United States under a PCIP, or plant controlled import permit. Similarly, local varieties have to go through a sanitation process. And these are very similar processes, except introduction implies introduction from a foreign source, or in some cases a source outside the regulating state, whereas sanitation refers to a local variety. The technology is the same and basically involves a series of therapeutic steps followed by extensive pathogen testing to confirm that the pathogen has been eliminated. Citrus has two well-established therapeutic techniques for virus elimination. One is thermotherapy, which, as it is implied by the name, involves virus elimination by heat. This is a commonly used technique in many different crops. The other one is more unique to citrus. This is shoot-tip grafting. In this process, tiny etiolated seedlings are produced in aseptic conditions, and a tiny portion of the apical meristem, approximately 0.1 millimeters, is excised and placed onto this tiny seedling. The principle here is that the very tip of this apical meristem has undifferentiated cells and consequently no phloem in which the pathogen can reside. So if successfully done, this can eliminate pathogens. Presently, most sanitized citrus bud sources or mother trees are maintained in protective structures. In the case of the United States, these protective structures are approved and inspected monthly by USDA-APHIS. In the case of the citrus gene bank, the subset which has been sanitized is maintained in one of these protective structures. The maintenance of this sanitized collection and the protective structure is very important or critical in allowing us to fulfill our mission of distributing citrus propagated materials for research purposes worldwide. I'd like to talk a little bit about today our cryopreservation program for citrus, which we are doing in cooperation with the National Laboratory for Genetic Resource Preservation in Fort Collins, Colorado. We have a APHIS-approved protective structure in which we house these sanitized plants. However, they are vulnerable to breaches, natural disasters, and other threats. So starting in 2012, we started cryopreserving these in liquid nitrogen in Fort Collins, Colorado. This provides a secure and safe backup for the materials in Riverside should some sort of calamity befall them. Our current collection of pathogen-tested propagated materials, which meets the USDA-APHIS standard for interstate movement of citrus propagated materials, currently numbers approximately 600. Of these, approximately 540 have been cryopreserved in Fort Collins, and we are actively working on cryopreserving the remaining amount. As future accessions are sanitized, they will be sent to Fort Collins when sufficient material is available. We're currently regenerating these cryopreserved materials and are performing research to determine whether any genetic or phenotypic changes occurred and to verify that the pathogen status has not changed. By doing this, we hope to secure these important genetic resources for the future users and also to contribute to the food security of the United States.